I am a biochemist by training, and I did my specialization in molecular biology, and then I stepped into glaucoma research in 1999. So basically I'm studying various cellular and molecular mechanisms that contribute to optic nerve damage and death of retinal ganglion cells, which are key events in glaucoma progression which contribute to vision loss. Most of my research is in animal models of glaucoma, and particularly in the Morrison's rat model of glaucoma. So we sclerose the outflow pathway, the trabecular meshwork, by injecting hypertonic saline through the episcleral veins. So essentially we're going against the direction of the aqueous humor outflow and causing scarring of the trabecular meshwork, which ultimately results in elevation of intraocular pressure. And this elevation of intraocular pressure can be maintained for various time durations, all the way from 2 weeks, 1 month, up to 2 months. But then by the end of 1 month, there is substantial damage to the optic nerve and death of retinal ganglion cells. So we use the 2-week or the 4-week time point to assess cellular damage, loss of retinal ganglion cells, and damage to the optic nerve. Essentially, the topic of the talk is elevation of endothelin receptors in glaucomatous rats. What we found is when we elevate the pressure in rats, when we elevate the intraocular pressure in rats, there is an increase in endothelin B receptor protein. So when that happens, it predisposes the retinal ganglion cells to cell death, and it also produces damage to the optic nerve. So we use either regular rats, wild type rats, or endothelin B receptor deficient rats. We elevated the pressure in one eye of these rats and used the other eye as a control, and we looked at changes. And what we find is that upon elevation of intraocular pressure, there is tremendous damage to the optic nerve in the wild type rats. There is also substantial loss of retinal ganglion cells, and these changes are attenuated in the ETB receptor deficient rats, suggesting that the ETB receptor is a key contributor to neurodegeneration in glaucoma, glaucomatous neurodegeneration. The long-term implications are that if you now develop certain drugs or certain gene therapy methods using adeno-associated viral vectors to knock down endothelin B receptor, hopefully we can have ways to protect the optic nerve from damage and improve the survival of retinal ganglion cells. So it's possible that if we work long term and develop a clinical trial, hopefully in addition to lowering intraocular pressure, patients can take a pill in terms of a drug that blocks endothelin receptors and promote neuroprotection in these patients. So this will enhance the vision in patients, because right now the current modality of treatment is mainly lowering intraocular pressure, and along the line in the future we may require both reduction of intraocular pressure and strategies for neuroprotection. There has been a lot of studies and a lot of candidates that have come along over the years. So the question is, how many of these would go to the translation phase and be used in patients? So it's to be time tested, and I'm pretty confident that ultimately there are 1 or 2 drugs that will hit the market. So the current limitations are that clinicians have to do visual field testing, and that's very long and arduous. So hopefully we can come up with ways to assess the retinal ganglion cell survival and integrity of the optic nerve. We should have better means to assess that, and then we can have progression to neuroprotective drugs. I would hope that over the next few years we develop some key candidates and drugs that will be available for patients for neuroprotection, and to take one step further, which is even more challenging, we hope that there are strategies for regeneration of the optic nerve and making synaptic connections to the brain, the higher order brain structures. Those are the key issues, and they'll be needed because as we have the aging population and greater incidence of glaucoma, we'll definitely need those approaches to restore vision in patients who have lost eyesight.